Biology 0610, Paper 6, ATP, Variant 1, May June 2024. Question 1. Cooking fruit and vegetables in water reduces their vitamin C contents. The vitamin C is transferred from the fruit and vegetables to the water by diffusion. DCPIP can be used to estimate the concentration of vitamin C. The blue DCPIP solution reacts with the vitamin C solution and becomes colorless. So its color is changed from blue to colorless. So a student investigated the effect of cooking time on the vitamin C content of the water. A piece of foil was wrapped around a beaker so that the base and sides of the beaker were covered. The top of the beaker was not covered. So beaker was wrapped from sides and from the bottom. A citrus fruit was also kept into it. So hot water was added. So hot water was added into it so that the piece of citrus fruit was covered. A stop clock was started. Stop clock to measure the time, to calculate the time. 1 cubic centimeter samples of the liquid in the beaker were taken every minute for 5 minutes. After every minute. After every minute for 5 minutes. So this duration of this investigation was for about 5 minutes. Each sample was put into a separate test tube. A 5 cubic centimeter syringe was filled with DCPIP solution. Any air bubbles present in this syringe were removed. So they were removed. 1 drop of the blue DCPIP solution was added into this test tube containing the first sample. The test tube was shaken for about 5 seconds to mix the contents. The color of the sample in the test tube was observed. The student repeated step 7 until the sample remained blue. The volume of the DCPIP solution remaining in the syringe was recorded. The syringe was refilled with 5 cubic centimeter of DCPIP solution. Step 7 to 9 were repeated with the rest of the sample. So for every sample, the same procedure was adopted. Now at the end, the figure 1.1 shows that volume of DCPIP remaining in this range and the volume of DCPIP added into the range at that time, which was added into the sample. So in the first minute, it was 3.6 and remaining 1.4, added 1.4, and remaining 2.8 and added 2.2, remaining 2.2 and added 2.8. 4 minutes, 1.8 and 3.2 and 1.6. So this value is not given. This is missing value. We have to calculate this missing value. On the next page, the formula has been given for this. So we will move towards the next page, and on the next page, we have to calculate the value or this missing value. You can see the starting volume of the DCPIP added. It was for 5 minutes. So the volume which was taken at the start of experiment, it was 5 cubic centimeter. So the volume of DCPIP remaining in this range, now it is 1.6. I have taken this value from the figure which has been given on the last page. So when we calculate this value, so the answer will be 3.4 cubic centimeter. So the results are 3.4 cubic centimeter. Now, it means in the last minute, 3.4 cubic centimeter volume of DCPIP was used. And it means the as the time is increasing, the volume remaining is decreasing. So now we are going to the next question, and in next question we have to make a table, prepare a table and record the results shown in figure 1.1 and your answer to 1. So we are we will draw or construct a table in this way. The first column will show the time and the second will show the volume of volume of DCPIP remaining, or volume of DCPIP used. So the time was for about 5 minutes. 1, 2, 3, 4, 5. So in this way, we have to write the all values which were given into the figure 1.1. So 1 point in this way I'm writing. You have to follow the same instructions in, and they have given all instructions. So it is very easy to solve this paper. So be careful while you read all instructions given. So here you can see the last value which we had calculated. It was 3.4. Now we will move towards the next question. So the next question is state a conclusion for the results. And you can see students in this case, as time is increasing in the downward direction. As time is increasing, the volume of DCPIP used is also increasing. It is also increasing, and the volume which remained behind, it is decreasing. It is decreasing, and the volume remaining is decreasing. The volume used is increasing, and in same, the time is also increasing. As time is increasing, you can say in this way. So we will write this conclusion in the given space. So the state a conclusion for your results. So, and we can write in this way. You can say, as time is increasing, more vitamin C is diffused out of the food. It means more a volume of DCPIP is used to make it colorless. So in this way, there is more diffusion of the vitamin C out of the fruit as time is increasing. So it was conclusion or it was a result. Now we are moving towards the next question. And the next question is state the independent variable in this investigation. So, the independent variable, which is always taken on x-axis, and it is here in this case, uh, it is the time. Because according to the time, the volume of the DCPIP is changing. as it is changed. So it means the volume of the DCPIP is the dependent variable, whereas the time is independent variable. So we will write over here time. Now the next question is, state the purpose of wrapping the beaker. Uh, as you know, if we wrap the beaker, it means it will reduce the heat loss. So it was the basic purpose. So we will write this, the purpose of wrapping the beaker was to reduce the heat loss, because the reaction will not be according to the correct results. Because if there will be more heat loss, the water will cool down very fast, so it will affect the results. Next question, explain why it was important to remove the air bubbles from the syringe in step 6. And you know, if there are bubbles in the syringe, so they can affect the volume which will be used for this purpose. So to maintain accuracy, accurate use of volume, or I think it's enough to maintain the accurate use of volume for this investigation. So it was all about question number 1. Now we are moving towards the question number 2. Okay. Question number 2, you can see a plan an investigation to determine the effect of light intensity on photosynthesis in an aquatic plant. So the aquatic plant can be used for this purpose. And here in this, you can see, students, first of all, you uh, have to plan in your mind, set what you are going to do in this investigation, what you should have to do in this investigation. So it will, when you will plan, then you will write properly. So first of all, you have to do that, you have to write the independent variable or dependent variable, decide which one is the independent variable and which one is the dependent variable. So after doing this, you have to focus on the apparatus which will use for this purpose. So you have to find out the effect of light intensity on photosynthesis. For this purpose, you have to take beaker, you have to take test tube, water, and above all, you need an aquatic plant for this purpose. So uh, you will have to set this experiment, and you have to maintain the accuracy throughout the investigation. You have to calculate the proper time with the help of stopwatch, and you have to maintain the intensity of the light. So our focus is on intensity of the light. So intensity of the light should be maintained. How it will be maintained? Because in this experiment, we need different light intensities. So the different light intensity should be maintained accurately. So for this purpose, you can use the table lamp, and the intensity can be maintained in this way, that the distance, you can change the distance. In the first, when you are going to give more intensity, you can keep the apparatus very close to that table lamp. And after sometimes, after the first, you can say observation, you can change the uh, place of that, or you can change, or you can increase or decrease the distance of that table lamp. So in this way, you can calculate uh, the effect of light intensity on photosynthesis, and then you can also repeat this experiment again and again to find out the accurate results. So in this way you can also remove those results which can be due to some errors, which are different due to some errors. So in this way you can plan an investigation. So I would like to write all this in this form. First of all, I'm going to write the dependent variable. In this case, the dependent variable will be the oxygen, which will be produced as a result of photosynthesis. So the independent variable will be there, because as a result of photosynthesis, oxygen is produced. So dependent variable in this case is oxygen. So for this purpose, we will we will set an apparatus. A large beaker will be used for this purpose, and in this way. This is for your convenience. I can draw this picture over here in this. A large beaker can be used for this purpose. And in large beak, you can invert a funnel. Under that funnel, you can have the water plant. And on the stem of the funnel, you can invert a test tube filled with water. So you can keep this into the light which you are going to conduct for this experiment. Okay, that in this way, it will be helped. You will set the apparatus and use a table lamp to provide light. The light intensity can be changed by changing the position of the lamp from the apparatus, set apparatus. When light is on, then start the stopwatch and observe the bubbles producing as a result of photosynthesis. The bubbles can be collected into the test tube. And these are, these are the bubbles of the um, oxygen. And in this way, this experiment can be repeated with the other light intensity. And one safety precaution which should be taken during this, you can make use of this, wear gloves or wear. I think one safety question is enough for this because it is very simple experiment. You have to save your hands. And secondly, this experiment can be conducted for about 10 minutes. For 10 to 20 minutes. So this experiment can be continued for 10 to 20 minutes. And after 10 to 20 minutes, you can find out the number of bubbles present or the number of bubbles. As you know, at low temperature, the rate of photosynthesis is low. When the temperature is high, the rate of photosynthesis becomes also high. So at low temperature, the number of bubbles which will be produced, they will be in small number, whereas the when temperature will be high, the bubbles will be produced in greater number. So in this way, we can make comparison between two light intensity, and in the two light intensities, the production of the oxygen or the process of photosynthesis will take place differently. So it was all about the question number 2. Thank you so much. Allah Hafiz.